Thank you. Okay, welcome everyone. We will continue now with the proteomics data analysis pipeline, second part. My name is Aivett Bilbao. I'm computational scientist at EMSL. This talk will start explaining why we care or we use mass spectrometry. Then we will revisit some concept about software for mass spectrometry based proteomics, as we'll revisit concepts that Yuchen already explained, and we will talk about some other free open source software tools. Then we'll transition to talk about multidimensional mass spectrometry data acquisition, talking about details of the data structures in the files acquired by the mass spectrometers, a specific processing software for data independent acquisition and ion mobility. And finally, we will talk briefly about top-down proteomics and interprotein analysis. In the flow of genetic information within a biological system, proteins and small molecules are essential to understand the dynamic changes at the phenotype level, particularly caused by external factors in the environment. Therefore, mass spectrometry, it's the method of choice. It's important for us because it's the method of choice due to sensitivity and throughput to study these molecules in complex systems. Synthetic biology aims to modify organisms to produce desired fuels and chemicals from renewable biological feedstock. And here in our lab, we are part of the Agile Biofoundry Consortium, and we are developing multidimensional analytical tools with ion mobility and data independent acquisition coupled with liquid chromatography for fast, comprehensive, and confident molecular characterization in the test phase of this development cycle. At EMSL, we have different mass spectrometry, cutting-edge mass spectrometry instruments that generate a lot of data. Many terabytes of data that it's impossible to analyze manually. And this is why we care about developing and using automated software tools, and these are necessary. Revisiting the discovery proteomics general pipeline, as Yuchen explained before, starts with the conversion and pre-processing of the data. Usually the data that comes out of the instrument comes in a proprietary format. And the typical tool or the widely used tool to convert this data to an open format that we can use with open source and free tools, it's a ProteoWizard. And typically we perform at least some signal processing, for example, the centroid, because the spectra is acquired in the raw spectra is in profile mode, especially from high resolution instruments of analyzing its intact molecules. Then we apply some centroiding algorithms. Then comes the step of identification. You heard from Yuchen about MaxQuant. Other software tools include MSGF Plus that David will be presenting later, and also MS Fragger and BibleSpec that basically compare theoretical or library MSMS spectrum with the experimental spectrum to do the peptide spectrum matches. Then we have tools that take those matches and perform a statistical analysis to assess and control the error rates of the faults, to evaluate the false discovery rates, calculate and evaluate. Follow it then for tools. There are other tools to perform quantitation depending of if it's isotopically labeled or with mass tags or label free. And then we have tools to perform a further statistical analysis like PMAR and MSStat, and then other tools to perform network analysis like Cytoscape. Here I would like to mention about the FragPipe. historically, the tools for proteomics have been developed in a step or isolated fashion according to the different steps in the workflow that you saw before. There is now an improvement towards making tools that are more comprehensive, where you can perform all that they are integrated, so they are more user-friendly. You can analyze your data in a more reproducible way. For example, the FragPipe, it has integrated the MS Fragger, which is the search engine to identify the MSMS, annotate the MSMS, and it has the philosopher tool to do the fault discovery rate analysis and other tools to do quantitation, including TMT and nitrate labeling. And also it includes the DIA umpire for analysis of data-independent acquisition spectra. So if you remember from previous presentations, we have been repeating this. The typical workflow: you start with the proteins samples, then you digest it into peptides, because these are easier to analyze. The mass spectrometry can better characterize these molecules that are smaller. The quality of the data it's better. They can be captured or characterized better by mass spectrometry, and we can apply the software tools. So let's talk about now how the data looks like in the file. But what comes out of the instrument, it's a file. So as you know, we have spectra, and the spectra is acquired by the instrument as the peptides are eluted from the liquid chromatography column. A typical representation is the total ion chromatogram, which is basically the sum of all the points at each spectrum, each time point. And then we can have peptides that co-elute when the samples are very complex. An overview of what is the difference between data-dependent and data-independent acquisition. In data-dependent, specific precursors are selected and isolated in a serial or sequential fashion. But in data independent, we have isolation of multiple precursors that are isolated in a systematic way, so we can build chromatograms for the fragment ions too, which is not possible when we acquire the data in data-dependent acquisition. Revisiting what the instrument is doing during the data-dependent acquisition, for each cycle, for each time point across the LC separation, the instrument is collecting first MS1 or precursor spectrum where there is no fragmentation, and then it detects the peaks, the precursors in that spectrum, and then sequentially selects one by one, and it's acquiring the fragmentation spectra of each of these precursors. Now, the limitations of this approach is especially visible, or has a high impact when analyzing complex samples, because the instrument has no time to select every precursor that is eluting from the chromatography column because of the complexity. So the approach is to select the top end, typically 5 to 10 most intense precursor ions during the elution. And then we have small isolation windows. If we think about the LCMS data in terms of time and mass to charge with the precursors that are being isolated, we can think of some precursors because that there is no time for the instrument and the separation to be selected. The disadvantage is that the high abundant ones, they get selected multiple times, and then the low abundant ones are missed. They are not detected. So then the reproducibility is limited, and some information is lost. Then what is a data independent acquisition consisting of? It's then we take those isolation windows, instead of doing them small for a specific precursor, we just open that isolation window and make to cover the whole mass range. In this way, we can operate the instrument, program the instrument to collect systematically fragment ion spectra across these isolation windows, and in this way create a fragment ion or digital map of all the signals coming out from that sample to characterize the sample. In this way, it's comprehensive, it's highly reproducible, and all, as I said, all the information is collected within a single run, and there is possibility of data remining for optimizing the results. The disadvantages are that the direct link between the precursor fragment is lost, and the data, it's much more complex. The DDA, it requires special algorithms and software. The typical tools for DDA will not work well with this data. And there is increased likelihood of interference due to the co-fragmentation, the co-isolation and co-fragmentation of multiple precursors within each experiment. There are several ways or schemes to acquire data independent acquisition spectra. As I mentioned before, the first stage is the precursor isolation. One of the first methods proposed for DIA was to just use the 2 experiments. One, instead of using several isolation windows, it used only one, like for example, MSE, it used only one experiment to collect data from all the precursors, and then the instrument, it's alternating between no collision energy or low fragmentation energy and high fragmentation energy to fragment then all the precursors within that time point that are eluting at that time point. More recent approach then proposed to use several isolation windows, so you can divide the precursor mass range into multiple isolation windows. Then we can acquire fragment ion maps for each of these isolation windows. This will reduce the complexity. This is possible with newer instruments that are faster. So it's possible to cover all the mass range with multiple windows in a time scale that is compatible with the liquid chromatography separation. In terms of the instrumentation, there are two main instruments. One is use a time-of-flight instrument, so it has a quadruple isolation window, and then the fragmentation cell, and then the time-of-flight type of detector. The other type of hybrid instrument also has a quadruple, but then has a device called C-trap and a higher energy collision cell, and it's connected to an orbitrap mass spectrometer. These instruments have different advantages and disadvantages each. Here we have an overview of the quadruple precursor isolation window schemes. At the top, we see the standard data-dependent acquisition scheme, where we saw that the isolation will be just randomly depending on the precursor that are selected. Then we have another approach that is called parallel reaction monitoring, where specific precursors can be predefined, and then the instrument, it's something between SRM and DIA, because the instrument is collecting spectra for the full spectrum for specific precursors that are selected. Then we have multiple isolation windows that can have all the same width. Or in D, we have a variable isolation, which it means that we can optimize the DIA method. And in the regions, in the precursor regions where we are expecting to see more precursors, we can then set these isolation windows with a smaller size. And then in this way, we reduce the co-fragmentation, co-selection of multiple precursors. And finally, another method that was proposed is to use staggered windows. It means they are shifted between consecutive cycles. This acquisition scheme helps to reduce interference too after applying the multiplexing step to generate the normal DIA spectra that can be processed with the typical tools. I was mentioning the similarities of PRM between SRM and DIA. You also heard from Yuchen about SRM and PRM. So the message here that I want to show you is now we introduce DIA compared to SRM and PRM. The upfront method development for SRM is very high because you need to select the peptides, you need to select the proteins, the peptides, then the fragments, the transitions, the collision energy. So that requires a lot of upfront method development. For PRM, you don't need to go up to the fragment or transition selection step. You only select your proteins and your precursors, your peptides. And the instrument is collecting a full MS2 spectrum for each of the peptides that are targeted. In contrast to SRM, where the instrument is just collecting one time point of the intensity of that fragment ion that was selected. So basically with SRM, we have chromatograms that are recorded in the instrument file format, the data. And for PRM, we have full MSMS spectrum with all the fragments that are detected. And then for DIA, we don't need to select, we don't need information about the proteins or the precursors that we want to analyze. We just set the instrument to collect and fragment everything that is coming out from the instrument. This implies that then inversely proportional, the data processing complexity for SRM, it's very easy because basically the instrument already did the job of making the chromatograms, and the researchers already did the job of selecting it upfront the proteins and the peptides and so on. For PRM, it's medium. Then we need tools for the data processing of PRM. We need tools that will create the chromatograms, and we'll find then from the data from the MS2 fragment ion spectra, we'll create the chromatograms of the fragment ions, and we'll decide which ones are the best for identification and to characterize those peptides and quantification. And then for DIA, finally, it's very complex. It requires processing, special processing, and we will talk more about in the next slides. The general workflow for processing DIA spectra, mainly we have 3. And the first one, the software basically takes all the DIA spectra and it performs an untargeted feature detection, and then deconvolutes or groups the precursors and the fragments based on the isolation profiles. Remember, we have chromatograms for the fragments, chromatograms for the precursors, so then we can group them according to the similarity of those profiles. For example, DIA Umpire is a tool that performs that. Then once this grouping is performed, the tool will generate a pseudo MS2 or pseudo DDA spectrum where the accurate precursor mask is assigned from this analysis of the deconvolution. And then we can take that output and use typical or standard search engines like MaxQuant or MSGF Plus or MSFragger. The next approach is more newer tools are being proposed now that are improving. Before, for example, FDARM were methods that you see here in the blue box, were methods that were very high computational complexity, so they were very slow, not practical. But now there are more recent implementations that can perform this kind of analysis. Means we don't need a spectral library to analyze the DIA data. For example, Cyclopedia has this pecan now, a new implementation that is called Wagner, that generates B and Y ions, or basically theoretical spectrum from the FASTA file. So we don't need a MSMS library to analyze the DIA spectra. And the PROCIT performs a deep learning analysis to predict the MS2, or to predict that library and then analyze the DIA spectra. And finally, we have the DIA-NN that also has algorithms to perform analysis without requiring a spectrum library. Then finally, we have the targeted extraction approach. In Cyclopedia, it performs, we can say it's something between library-free and targeted, because it also applies targeted approach to generate the fragment ions chromatograms in the analysis. But the more typical targeted extraction workflows utilize a spectral library that is generated in data-dependent acquisition mode and analyze it with the typical tools. So you can generate a high-quality spectrum library that will be used to generate for targeted data extraction. It means the software, when analyzing the data-independent acquisition spectra, the software can know in advance what is the retention time that this peptide is expected, and what are the fragments that are expected in the data, so to create the chromatograms. And this type of analysis also use a scoring scheme where you can compare the fragmentation pattern, basically the abundances of the fragments against in the DIA spectra against the library to increase the confidence of the identification. And basically, the widely used algorithm for the false discovery rate analysis in the DIA data, it's called mProfit, which basically calculates a score that combine also. It generates decoys from the library, DIA library. It generates also decoy spectrum that can be extracted from the DIA data, and also can be used to discriminate between the targets and decoys and apply a similar false discovery rate analysis. And this is the typical workflow for tools, for example, like Skyline and OpenSWAT. I apologize for this slide that is a little bit crowded, but it's a very nice summary of the development of data-independent acquisition methods and the tools that are available now. On the left, we can see the DIA, data-dependent acquisition schemes, in kind of a timeline. In 2000, 2003, 2004, these were first basically initial proof of concept where DIA was starting to be proposed. This was, for example, at that time, the acquisition was performed in 2 steps, because the instruments were not able to alternate between high and low collision energy to collect precursor and fragment ion spectra. But then in 2009 and 2010, Waters commercialized an instrument that was able to collect that precursor and fragment ion information within a single run. And then they released a software or that was proprietary to analyze the data. And it was until 2012 with the SWOT publication where the method, the DIA method, gained more momentum and more interest, because then the instrument was faster. It was able to collect more high quality information, and it was proposed this targeted extraction, targeted data extraction strategy similar to SRM, where we basically know we can create libraries from data dependent acquisition experiments to know what are the vectors that we are looking for in the DIA spectrum. And after that, several approaches have been proposed. Now we have different improvements in the instrumentation that can do a better isolation of the precursors, and now also approach that combine ion mobility. We have been talking and mentioning here Skyline, because it's a very important tool for proteomics, for mass spectrometry. And there are several reasons for that. The main one is because it's open source, it's free, and it supports many acquisition types. It supports SRM, DDA, DIA, ion mobility. It supports processing of all these kind of data. It supports different molecule types. It can be used for proteomics, metabolomics, lipidomics, and it supports the direct processing for the data that is coming out of the instrument directly. It doesn't require a file conversion step, and it's user-friendly. Okay, now we will talk about ion mobility. You remember this slide, which is how now here I have some small molecules design, but it's the same. They can be analyzed with the liquid chromatography to be separated, and then to collect the LCMS data. What we do with ion mobility, the instrument, then it has another stage that is in front of the mass spectrometry, and after the liquid chromatography separation. And what we can do is then add another extra separation step, another dimension in the data, that then we can further characterize and separate molecules that are not possible to separate by LC alone. So then instead of having one single spectrum per LC time point, we have what we call ion mobility frame, which is multiple spectra that that the instrument is collecting following the ion mobility separation. And also combined with this, the ion mobility separation, we can collect data, fragment ion data, by alternating high and low energy collision energy. And then this is then when we apply also data independent acquisition combined with ion mobility and liquid chromatography. And then the collision cell, it's activated to fragment the ions after the ion mobility separation. In this way, we can generate fragment spectra on a fragment ion mobility frame, where the fragments we have the same mobility as the precursor, and then we can apply these deconvolution algorithms to group them. There are several types of ion mobility instruments. The main measurement principles are drift tube ion mobility, the first one. All these instruments measure the drift of ions through a region filled with a buffer gas and under the influence of an electric field. The electric field will produce this drift of the ion and will allow this mobility separation. In drift tube, this drift field, it's constant and homogeneous. The electric field, the drift tube IMS, it's a technology that was developed by Dick Smith Group here at PNNL and later commercialized by Agilent Technologies. Then we have a traveling wave ion mobility, where the electric fields, then it's in a wave, it's applied in a waveform. We have trap ion mobility, where basically we have two stages, one of trapping and one of the elution. And the differential mobility analyzer that use a ramp of this applies a ramp of this electric field, and the field asymmetry finally that applies a square form to achieve the separation, the mobility separation. Why we care about ion mobility, besides adding this separation dimension that helps to acquire better quality spectra and characterize better the molecules. There is a property of the molecule that is called collision cross-section that can be derived from the ion mobility separation measurements. So the collision cross-section is a value that is related to the size and shape and charge of an ion of the molecule, and it corresponds, it's proportional, or is related to, of course, to the area that collides with the drift gas. It's basically this surface around the molecule. It's a robust physical chemical property, can be compared between labs, and it depends on the drift gas that is used in the mobility cell. There are basically two methods to calculate the collision cross-section. The step field method on the top, that is the only method that allows, it's commercially available to allow measuring the collision cross-section without using any internal ion as calibrant. So basically the molecule is measured in different different step values of electric field, and then a linear regression can be performed, and an equation of a principle called the Mason-Sham equation can be applied to calculate this property directly. And other methods, especially used in traveling wave ion mobility, or what we call single field ion mobility in the drift tube instrument, basically requires calibrants. Ions are measured under the same condition, and we have no collision cross-section values, and we use these to calibrate all the rest of the ions, the features that are detected within the data. There have been methods that have been published to predict the collision cross-section. One of the most recent ones is predicting the collision cross-section from peptides using deep learning methods, and basically takes the peptide sequence and it applies deep learning and generates the collision cross-section of that peptide sequence. The existing LCMS tools are being updated to process ion mobility data. One of the first ones was Skyline. Here, it's an example. You can see a calibration curve when you have multiple, a dilution experiment with multiple concentrations. Molecules is a mixture, peptide mixture measured at different concentrations. And on the left, we have only the quantitation made without ion mobility, and on the right, when we put also ion mobility combined with the LC, and you can see that when we add ion mobility, we have more, the limit of quantification is lower, because Skyline was adapted to then filter the chromatograms, the LC chromatograms, by using only the mobility region that corresponds to that molecule, and we can see how it decreased also the interference. Another tool that was adapted to process also ion mobility data was OpenSWAT. Basically, they extended the model that they implemented for SWAT or DIA, LCMS, typical standard DIA, and they extended the same scoring model by adding properties now that take into account peak shape and properties from the ion mobility dimension to compare these against libraries again. Here now I want to mention the PNNL preprocessor tool, a tool that we developed here at PNNL in collaboration with Agilent Technologies. And it's a tool that it's implemented with a series of algorithms that allow to process or apply signal processing algorithms to the raw spectra that is coming out of the instrument to facilitate and this enhance significantly the results for all the downstream processing analysis. We have different algorithms for data compression, interpolation. We have ion mobility demultiplexing, which we will not cover here, but basically it's a methodology to acquire data when you can program the mobility to be encoded by multiplexing ion packages, and then we apply a software algorithm to demultiplex this data. We have the multidimensional smoothing and low intensity filtering. Other algorithms are called spike removal to remove all other type of low noise, saturation repair algorithm, and we also have options to export metadata. Here on the right, you can see an example of how it can improve or facilitate the peak detection for non-targeted feature detection in the raw spectra. After applying smoothing, we can see that the algorithm can better, the peaks are better defined, and the algorithm can do a better job to detect the real signals. Finally, for the ion mobility, I would like to show you about SLIM, the structure for lossless ion manipulations, ion mobility technique, which is basically traveling wave based, and was developed here also at PNNL and commercialized by Mobile Ion Systems. I think it will be released in the next few months. It provides the highest reported ion mobility resolving power. Here we can see on the left the resolving power typical for the drift tube instrument. It's 50. When we apply SLIM, we can separate up to 300. And this is very important, for example, to study and separate isomeric molecules. Here on the left, you see an example of phosphopeptides that they basically have the same precursor mass. So they won't be able to be distinguished by LC because they have the same LC time point. They are positional isomers. It means the chromatography properties are the same, so they will coelute. But then if we perform ion mobility with SLIM, we can separate these molecules and resolve them. Now our final section, it's about top-down proteomics and intact protein analysis. The advantage of top-down proteomics is that it allows the specific characterization of proteoforms. When we analyze the intact protein, it means we are not digesting the protein. Then we can tell from where is the position of specific modifications if we have multiple proteoforms, multiple proteins that can be modified in different positions. That in the case of applying the typical proteomics bottom up, when we digest the proteins into multiple peptides, if these proteoforms have shared peptides and they have shared modifications, once we do the digestion, we won't be able to tell from like exactly which proteoforms they are, because there are many possibilities. So that's the advantage of top-down and the importance of doing this kind of analysis. There is a top-down proteomics. As I say, we don't apply digestion, we just analyze the full proteins, and we can fragment the full proteins. There is middle down, where there is a limited digestion, and the typical, compared with the typical bottom-up experiments. The intact protein analysis is basically what we call is the analysis at the MS1 level. There is no fragmentation. When we are analyzing the intact protein, the masses are very large. So the algorithms that need to be applied to process this data are very different compared to the bottom-up proteomics approaches, because instead of detecting now peptides when we have isotopically resolved ions, here, because of the size of the molecules that are so big, then we started to detect charge envelopes. As you can see on the top, there is only a single peak to detect this intact virus, and we cannot resolve the charge states. And then here is where the mass resolving power is very important. As you can see in the next two figures, when we use instruments that have more higher mass resolving power, then we can distinguish this charge, the charge envelope baseline resolution. These instruments, these instruments are commercially available or recently modified. We have here instruments that for FTI, for Fourier transform ion cyclotron resonance mass spectrometry, the highest resolving power with a 21 FT or Tesla, 21 Tesla. So it can resolve better the charge envelopes to detect these big, large molecules. And for example, you can see here the comparison when we use the 21T, we can detect many more or most all the proteoforms with the sub-ppm precursor mass accuracy. And this allows the detection, more confident detection and distinction of the proteoforms. Finally, I would like just to mention this consortium for top-down proteomics, that it's compiling and describing the tools, the software that are used for top-down proteomics, if you are interested to perform this kind of analysis. And now I'm open to answer questions, and I thank you for your attention.
Thank you, Aivett. Some of those graphs, they took very complicated processes and put them into pictures. Those are great. I'll have to steal some slides from you. It's at noon right now, but let's go ahead and go through some of these questions. Some of them you answered along the way. Let me start with one of the ones that just came in through the Zoom chat. So top-down proteomics is based fully on in vitro analyses or both in vitro and in vivo analyses. So what type of samples can feed in there? Something that's complex or something that's more a true in vivo sample?
There are specific sample preparation steps that should be applied in each case, but there are top-down is an active area of research now, and a lot of protocols are being developed. You can study the intact protein, what is called denatured. It means like closer to in the structure that it should be in the in vivo, or you can try to isolate the protein and detect it denature. So there are protocols that are different. I think both, I'm not 100% in vitro, but as close.
Yeah.
There are
So when
methods
you're
to make
going
it.
to those native measurements, yeah, if it
Yeah.
keeps that secondary conformation, then you could even see that ligand binding or other really cool things that could be more of a true in vivo system. One question that came up when you were on your day IA slide, can the pipeline be automated or does it require user inputs from a GUI or parameter choices?
There are both type of tools. There are some that you can just basically select your settings, prepare your settings files, and just start running as see if you prepare your setting files correctly. You will just wait and it will be done. So yes, nowadays we have tools that can be run automatically.
Great. And I think there's other ones that you can answer over the day, but one other thing is someone asked about proprietary software, and you mentioned a lot of software tools that, are made in our community that kind of get around that. So I think in the mass spectrometry community in general, they're constantly making tools so that we all can utilize them. And I think that was one thing for both you and Yu-Chin, trying to show those softwares that can be used for these types of analyses that can be downloaded and used for lots of different applications.
The proprietary tools are typically much easier to use, but they are slower. They are coming behind the new developments. The new algorithms typically comes from academic software. So it's an open source software or a method that have been published that it takes time for industry or for the software vendors to implement them. So yeah, that's the trade-off. Vendor software, besides being expensive, you have to pay for them. They don't have the latest.
Yep. Okay. Well, I think
And
with
I'm happy to answer any question that anyone can have. Just send me an email.
Yeah, there are a few more on Discord too. But we'll get through all of them today. So
Thank you.
Thank you, Aivett. Leanne, do you have any closing remarks or anything?
Nope. That was great. I will have this afternoon with the students in tutorials, and then tomorrow morning, once